Next up, we have Bryan Briney coming from the Scripps Institute in San Diego. And he's going to tell us about some work he's done doing some very, very deep characterization of a few individuals.
All right, hello again, everyone. This is actually already published, so I'm not just going to like read you the paper back. That's not very exciting. So I think part of the idea was to talk about analysis tools and stuff. So I'll talk more about how we're doing the analysis more than like what the biological results are that we got. So that's kind of the focus, even though we're going to essentially walk through the slides from the paper, walk through the figures from the paper. So talking to people here, I think there's also been a consensus from a lot of people that I've heard is that why do we need to sequence billions of these things? Like what's the added value beyond a normal MySeq run or something? Why do we have to do more? And Jamie mentioned it especially, what's even the point, right? You need to have a point to do something this big, right? So we kind of had a point, which is why we did this. So our group at Scripps works on a concept called reverse vaccinology. So HIV vaccines are extremely difficult. Most vaccines that you get are essentially mimicking the virus infection. So you have killed virus or an activated virus or proteins from the virus that you inject into your body. Your body thinks it's being infected, mounts a huge response, including a memory, and then you're ready next time the pathogen comes. So that doesn't work at all for HIV. There's been no success kind of mimicking viral infection. And that, in retrospect, kind of makes some sense, because if you actually get HIV, you don't clear the infection. So mimicking the HIV infection doesn't result in a protective response, right? So in retrospect, it's easy to say that, but people didn't expect that necessarily. So what we do instead is identify really good antibodies, protective antibodies the kind of antibodies you like to make by vaccination. And instead of starting with the virus, you start with the antibody and work your way back to some sort of vaccine immunogen that will specifically target the early germline precursors of those antibodies and then drive them towards in like a maturation trajectory that leads towards these broadly neutralizing antibodies, right? So essentially you're going from point A to point B. Point A is the germline antibody, point B is this highly mutated broad antibody. So after like decades of work that I wasn't necessarily involved in, a lot of these super broad protective antibodies were discovered, and they're rare and they're very hard to find, which is why it took so long. So we have a bunch of point Bs. The point of this experiment is to find out what all the point A's are. You want to go from point A to point B. What do all the starting points look like that we could hopefully elicit and drive towards these broad antibodies? So what we did was we took leukapheresis, which is basically as deeply as you can sample the peripheral blood from a person. You hook them up to a machine, and it's essentially a continuous centrifuge. It pulls out all the white blood cells, right? And then it puts the blood back so that they don't like shrivel up like a raisin. But it allows you to take essentially all the white blood cells in the peripheral circulation. Then we amplified those, sequenced them, and then there's lots of computation that happens after that. And Victor, I think, put something similar to this up a few days ago. It was important to us because we wanted to look at diversity and things in the repertoire, and also we wanted to make sure the things we were doing were reproducible. For each one of these leukapheresis samples, we split it into 6 biological replicates, so separate aliquots of cells, and then processed each of those independently in triplicate. So we isolated RNA from this sample separately, took separate RNA samples, amplified them, and sequenced them separately. So in that sense, there's a couple of things we can look at here, is that we should see essentially identical results between these two groups, right? This is the same pool of RNA that you're sampling, so you should get really reproducible results. But also, in terms of diversity calculations, most diversity metrics leverage what's called repeat observations. So it's hard to know in any given sample, even if you use molecular barcodes, if you see the same sequence or the same clonotype more than once, even if you know it was 2 different RNA molecules, you don't know whether it's 2 RNA molecules from the same cell or from different cells. And I would consider repeat observations in terms of an antibody sequence 2 different cells, right? If you see 2 RNA copies from the same cell, I don't know if you'd call that a repeat observation from kind of like a diversity standpoint. So what we can do with this data then is, if we see something in this sample and then again in this sample, we know for sure that it came from 2 different cells because it had to come from 2 different cells, right? So we got a lot of sequencing reads. The raw reads are about 3 billion total. After correcting with barcodes and stuff, we ended up with about 300 million reads that were kind of, so again, in terms of barcode correction, we didn't do the 3 or more reads. We did like 1 or more read in each barcode bin. So we're not necessarily error correcting, we're just bias correcting. So if we saw 10 copies of the same RNA molecule in our sample, we collapsed that down. But if we also saw 1 copy of the RNA molecule in our sample, we kept that also. So the error correction is not necessarily coming from the barcoding. So I'm gonna breeze through a little bit here in terms of repertoire uniqueness. So at a high level, and these are not really showing, well, there's actually supposed to be lines here on these. So good thing this is published, so you can actually follow along with the paper if you'd like to. So I don't know what happened here, but regardless, at a high level, we see pretty similar repertoire. So this is the V gene use of all 10 subjects on the x-axis or on the y-axis, and the V genes going along on the x-axis. So you can see as a general rule, the things that are most common among one subject are most common across all the subjects. But what we can then do, I think none of the lines are showing up. So again, I'll draw them with a pointer. There are like sigmoidal curves right here, and they're multicolored. They're very pretty. Let's see. Did it not come across properly? There we go. Fantastic. I told you there were lines. All right. Very good. Thank you. Saved it. So again, so everything's fairly similar. The CDR3 lengths of the 10 subjects overlay almost perfectly, right? So there's not any super high level obvious differences. But I told you there were sigmoidal plots there. So what we then did is, using increasing numbers of sequences, we calculated what's called the Morrissey-de-Horn's similarity index. And again, I think Victor mentioned this earlier. This is the math behind it, but the idea is it's a way to calculate the similarity of populations based on the frequency of their species. So in this case, the species is what we call the VJ CDR3 length. So we collapsed every sequence down to just its V gene assignment, so like VH12, the gene, not the allele. VH1, so that way we'd make things similar across people, right? We didn't want to have alleles be the reason they were different. So we've collapsed each sequence down to just the V gene assignment, the J gene assignment, and the CDR3 length, and then compute the frequency of all of those things. And that distribution of frequencies becomes our set of species, right? So if the VJ CDR3 length is a species, we count the number of each species. And those distributions can be compared between 2 populations. So a couple of interesting things that we think are important. So we wanted to sample at a variety of depths. And the reason is because if you imagine like a big bag of fruit, right? Even if the 2 bags of fruit are identical and they have like 1 million pieces of fruit, like 1,000 different kinds of bananas and a bunch of different kinds of apples, if you reach in and grab 1 piece of fruit, the odds that those 2 match are basically 0, even though the bags themselves are identical in composition, right? So we should see something like that. We should see basically no similarity at shallow sampling and increasing similarity, and then hopefully, we had hoped, a plateau at some point where you realize you've sampled deeply enough that increased sampling doesn't give you more information about how similar these repertoires were. And that's what we saw. So in this case, if you compare one subject to themselves, that's the red line, at about 10,000 sequences or so, you're essentially indistinguishable. So Morrissey-de-Horn similarity goes from 0 to 1. 0 means not similar, 1 means perfectly similar. So by about 10,000 sequences, comparing a subject to themselves results in a Morrissey-de-Horn similarity of about 1. And different subjects kind of plateau at about 0.7, 0.8, right, in that range. So those subjects are distinguishable from each other, which is good. And again, it's important to us that these plateaued, right? If we had kept sampling, these lines are going to keep coming up here and eventually get there. So we were sampling deeply enough to distinguish these repertoires. So next we constructed what we call, but what it actually is called, a one versus rest or one versus all SVM classifier. So SVM is a support vector machine. I'm sure some of you are familiar with this, but a brief review for those that aren't. The idea is you essentially have points mapped into space, and the space can be lots of dimensions big, but the human brain can really understand 2 dimensions pretty well. So if you imagine an XY plot and there's a bunch of dots, you try to find a line that best separates those dots into the appropriate groups. So if you have height, for example, and you have wanted to define people by their country of origin or something based on their height, right? So if you had some sort of line that could group those two dots into one country versus the other country by plotting height, that's the idea of a support vector machine. You want to draw some sort of what's called hyperplane that best separates two populations. Then what you can do, and that hyperplane is super important, it's called a maximum margin hyperplane, and what you can then do is with new data, you plot that new data and you can predict what population it belongs to based on where it falls relative to that line, right? So what we're doing here is, again, this is important from our biological replicates. We had 6 different biological replicates. So what we can do is hold one of those out for each subject. We can train the model using 5 of the 6 biological replicates and then test it with the 6th. And the idea is, if we give this model a distribution of VJ CDR3 lengths and tell it what subject it's for 5 of the times, then we give it a new subject, a new repertoire, and say, which subject does this belong to? And if it guesses right, or I mean, predicts is maybe the better word. If it predicts right, great. And this is not a great way to measure it, but one way to measure that kind of correlates roughly with accuracy is the area under the receiver operating characteristic curve. So that's what's shown here. So what we can see is that by about 500 sequences, we're basically getting it right most of the time. So using 500 sequences, you construct just the VJ CDR3 length species population. You can now say with reasonable certainty which subject that repertoire came from. Now, again, we talked about data privacy and all that business before. This is like 10 subjects. If we had 10 billion subjects, the odds that we could do this reliably is going to go way, way, way down, right? So also, we had to do a lot of sequencing on each one of these subjects to do it. So this, I don't think, should cause people concern that we're going to be immediately able to like fingerprint people by their antibodies, right? And again, one other thing that we haven't done is if we took one subject 6 months later, would they even look like themselves, right? That's another question to look at as well. Anyway, what we can say though is that the repertoires are distinct enough at this one snapshot in time that we're able to identify who is who just based on the repertoire. So I'm gonna breeze through the diversity stuff because it's not necessarily, I think, interesting in terms of our methodology, but one of the things that we did use here and for diversity and for the rest of the talk is we use clonotypes as kind of a proxy for sequences, because since we use barcoding but we didn't do error correction necessarily, we took things that had only one read in the barcode bin, we wanted to make sure that sequencing errors didn't dramatically influence our diversity calculations. So our definition of clonotype that we used here is kind of what people use in the TCR world, which is the V gene assignment, the J gene assignment, and the same CDR3 amino acid sequence, right? So it's not like 80% homology in the CDR3, it's the same exact amino acid sequence in the CDR3. So what we did is we collapsed all of the clonotypes into clonotypes for all of each of the individuals. And what a rarefaction curve is, for those of you that aren't familiar, is you sample your population at increasing depth and you count how many things you saw that were unique. So if we had completely sampled the population, this line would start to come up and tail off and become horizontal. What that means is you're sampling more and more deeply, but you're not seeing any new things, which means you've basically seen everything there is to see in the population. So you can tell we have not even gotten real close to that at this point. So there's a lot of clonotypes there. And even at this depth we're sequencing, we're only just kind of starting to bend the rarefaction curve. So we used that same data to then calculate or estimate diversity. So again, this is where the repeat observation thing comes in. So there's 2 estimators that we used, Chow and Recon. And again, Victor talked about these if you were at his data analysis talk. But so Chow, I won't go into the details, but the idea for both of these, essentially, from a non-statistician talking, is that you can, using the number of things you saw once and the number of things you saw twice, you can statistically infer the number of things you saw 0 times, which is the things that were there but that you didn't measure, right? So that gives you a way to estimate species richness, or the number of things that were in your population. So there's a couple of things that we found interesting about this plot. So again, we did that with increasingly large samplings of each subject. So as a general rule, Chow and Recon agreed. They were easily within an order of magnitude. I think they were within about a factor of 2 of each other, which is encouraging that the two estimators were getting to approximately the same answer. The other thing that we noticed is these lines should be exactly horizontal, right? Because the amount of diversity doesn't change depending on how much we sampled, right? The amount of diversity that was there should be the same no matter how deeply you sample, but increasing sampling led to increasing diversity estimates, which suggests that there's more very rare things in the antibody repertoire than these models expect, essentially. So these models are potentially underestimating the diversity because they underestimate the number of very, very rare things that are in the antibody repertoire. So one last thing is that we then pooled our subjects. And again, someone talked about this earlier, raised it as a question somewhere. What do you do if you sampled subjects at differing depths, but you want to kind of look at the diversity of the groups as a whole? We thought about that and said, whatever, let's just use all the data, right? So we've used different amounts of data for each subject. So that is a caveat here. What we did was we took unique sequences from unique clonotypes from every subject. And then for every combination of 2, 3, 4, all the way out to 10 subjects, we pooled the sequences and then recalculated the diversity estimates both with Chow and with Recon. So again, as you can see, as a general rule, they tend to overlap pretty well in terms of the estimators agreeing. But one thing that I found especially interesting is that, especially for Chow, but for Recon tails off also, is that I had expected this to be much more linear, right? You're adding more and more different people, and you don't seem to be increasing the total diversity contained by those people. Once you get past about 6 or 8 subjects, you're kind of leveling off there. So the metaphor we like to use to describe that is if everyone's antibody repertoire that they have in their body is like a bucket of water, right? So there's much more diversity that you can make than you have in your body at any given one time. There's not enough cells to have every possible antibody, right? So the idea is we're not necessarily all scooping our diversity from different pools. So there's this huge swimming pool full of possible diversity that our BCR repertoire can make. And we scoop out one bucket, and that's what we hold in our body at any given time. What this suggests is that we're all scooping from the same pool, right? We don't have our own separate pools that we're scooping from, right? Because as you add more and more people, you're not necessarily adding more and more diversity beyond a certain point, which I think from a vaccine perspective is super exciting, because if we're all drawing from the same pool and we're trying to target specific antibodies, that suggests that it's more likely that everyone has something similar, that we're not all markedly different in that respect. So one last little part is sharing between people. So we made, again, using clonotypes, we made every combination of 2 or more people and calculated the frequency with which clonotypes were shared. So this is kind of interesting kind of computational issue. So clustering these things gets difficult, right? So if you have 300 million sequences, running like CD-HIT or vClust or something is gonna take you a while. So we looked at a couple of things, and Ari's probably going to shake his head in shame. We looked at Spark and we didn't know how to do it very well, so we didn't use it. The thing that we found that was by far the fastest, if any of you are familiar with Linux or Unix, Unix sort with unique is crazy fast. So it took like, we started running like a pair of subjects that had like 150 million reads, and it was taking days using CD-HIT, and it took like 8 minutes using sort unique, right? So sometimes the simplest things are the fastest, right? So anyway, so that's kind of an interesting, so don't necessarily knock those small little command line words. Those are very, very powerful things. So what we found is that all 10 subjects shared about 1 in 4,500 clonotypes, right? And I thought that was shockingly high. I would have expected almost nothing. I would have expected almost no overlap between people. So unfortunately, my gut feeling was considered by reviewers not a valid statistical estimate. So they wanted something more kind of grounded in actual evidence. So when we say more than we expected, there's something other than my feeling. So what we did was we used Igor or Igor, I don't know what we call it, but I call it Igor, to infer or to generate synthetic sequences using their default recombination model. So what we did here is we generated about 3 billion sequences total, or about 2 billion sequences total, 1 billion using AIRR's default model, and 1 billion using models that we inferred for each subject. So we inferred VDJ recombination models for each of our 10 subjects using naive B cells only, so unmutated IgM B cells. So what we found is that using the default model, which was generated using non-productive rearrangements, so these are under no selection, both peripheral central tolerance selection and also selection in the periphery. What we found is that there was far less sharing using that default model. What that suggests, so the black is the subjects we actually observed, red is the default model in AIRR based on just non-productive rearrangements. So what we found, and what we think that means, is that the reason we have sharing between people is not because VDJ recombination accidentally or coincidentally makes the same thing over and over again. That's not the reason, because we're seeing, an order of magnitude or more sharing than we would expect based on just coincident VDJ recombinations. What's interesting, though, is we see far more sharing in the naive repertoires, the synthetic sequences built from VDJ recombination models of naive repertoires. So that's somewhat strange. So what we think might be happening is a model that looks something, one more thing before we go. But what we also notice, again, from repertoire similarity measurements, is that the IgG, the class switched repertoire, is far less similar between people than the naive or the overall repertoire as a whole. So again, another thing that people have suggested, myself included, was that it's not that VDJ recombination coincidentally makes the same sequence, it's that exposure to common antigens drives the convergence of your memory repertoire. So we make the same antibodies because we've seen the same pathogens. This suggests that that's not the case, because the pathogen-specific repertoire, the things that should be the most similar, are in fact the least similar. So what we think is happening instead, because the naive repertoire is extremely similar between people, is that you have this model that looks something like this, where you, at the original VDJ recombination, the similarity is very low. You have massive diversity and everything's super different across people. But after selection during early B cell development, essentially central tolerance, negative selection against self-antigens, selection for productive binding with a light chain and the surrogate light chain, all of those processes kind of homogenize everyone's repertoire. So at that point, the point that the repertoire leaves your bone marrow, we are at our most similar. And then once you're exposed to pathogen, you then kind of diversify, sometimes convergently with other people, but in a lot of times divergently from everybody else. So even if you are exposed to the same pathogen, and I think there was some work that I forget who showed it earlier, that even haplotypes potentially can drive responding to different parts of different pathogens, right? So there's definitely evidence that our responses to pathogen are not in fact more similar than the naive repertoire, but are in fact less similar than the naive repertoire. So that's the end of the talk. And so hopefully we won't have to put this part of the slide up here anymore, because it'll all be like in AIRRC4 or in NCBI or something that we all have access to, and we don't even have to really mention it because we all know where it's going to go. But all the code we used is on GitHub. You can use it for free. It's all open source. The raw data is in the SRA, and the process data, as well as all the code we used to process the data and make the figures and everything else, is on GitHub as well as a series of Jupyter notebooks, if you'd like to do that. And also, if you're interested, it's going to be probably, if you're computationally savvy, it's probably going to be a little bit remedial, but I'm giving a tutorial on AbStar and AbCloud and all of these things. That's also on GitHub. So it's just a Jupyter notebook that walks you through kind of getting started with the process. That's also on GitHub. So if you weren't able to make it to the tutorial, you can do it at home. And lastly, just a couple of acknowledgements. We had a summer student who was fantastically productive, as well as Colin Joyce, who I believe is here somewhere, a graduate student. And our clinical partners are HemaCare, and then of course the funding is important, because without that we can't do any of this. So any questions? Oh, we can just share. I guess we can't, never mind.
So can you go back to the slide with overlaps?
Yeah. That one, this one?
Yeah, this one.
Yes.
So, I mean, have you ever considered that it is possible that a synthetic repertoire generated by default by AIRRC was learned on some data, I think, produced by Adaptive, and they have a completely different VJ usage profiles from your actual data set. So just because, I don't know if you have sequenced with Adaptive, but maybe you've sequenced it with your own protocol. So as you define a clonotype with V and J, and if you shift the variable joining usage, then you will get like less overlap. And then if you will train to feed the actual usage better, then you next with subject-specific models, and perhaps there will be only a single, you don't need subject-specific model, you just need a model for your entire data just to even out the difference in V usage that come from methodology. You will get like more or less moderate overlap. Have you checked this?
Yeah, so one nice thing about AIRRC that I really enjoy is that they give you ways to essentially determine how two repertoires differ. So there's synthetic repertoire versus like an inferred model, right? So it turns out that most of the differences are in the CDR3 area. So whether it's the amount of trimming or the amount of N addition or the D gene usage, so it's not necessarily dependent on VJ usage. So the different primers in theory shouldn't make a massive difference in that area. So most of the differences came from what essentially is the VDJ recombination model that AIRRC produces. They make a lot more long CDR3s, and it's a lot more diverse in terms of nucleotide. So I didn't have this slide up here, but if you look at the nucleotide composition of CDR3s, the things that are most shared and the repertoires that we observed are much more restricted in their amino acid content of the CDR3s. So you're essentially losing hydrophobic and highly charged amino acids, and you're settling on a huge enrichment of like polar, non-charged, kind of neutral things. You know, lots of tyrosines, lots of glycines, that kind of stuff.
Yeah, right. That's really interesting because, well, I was always wondering if there are actual some differences in, as it was previously claimed, there are almost no differences between the rearrangements that observed in different people if you look at the naive cells, but it looks like there's a huge difference, and the description is really interesting.
Thank you. This is such a great study. And I was wondering, because you were talking about your repertoire changing over time, have you looked at the same donors over multiple time points, and how much variation is there?
Right. So we just got, I think, most of these 10 subjects back a year later. So we're sequencing them now. So we'll see. And that's super interesting, right? So if you look at the similarity between repertoires, are you maybe not perfectly similar to yourself, but still distinguishable as not somebody else? Or do you get so dissimilar that now you're as similar as somebody else would have been, right? Yeah, that's super interesting.
So presumably, certain things that are expressed at really high levels, you're always going to see them, I guess, at the same high levels. But rare clonotypes, I mean, that's where most of your change is going to be, maybe.
And we'd also expect a fair amount of change in the naive repertoire, because that seems to turn over a lot more and is less. I mean, you get a lot of turnover during acute infection, but the memory repertoire, I think, studies have shown kind of stays a little more constant, but you're churning through your naive repertoire, and there's so much possible diversity there that my guess is that's where you'll see a lot of changes.
So wait with bated breath.
But there's a lot too, like we're looking 1 year after. What if you looked every week or every month for 6 months or a year, right? How is the change kind of gradual, and you can kind of see a slope, that kind of stuff. There's all kinds of cool stuff to happen there, yeah.
So you have been looking in periphery. What about bone marrow? How do you expect that to change or to differ between individuals? Because I guess that's really what matters in terms of protection.
Yeah, so you're talking about plasma cells. So that's going to basically be your serum antibody, right? Yeah, so we've started studies on that too. So we wanted to do not this depth. I mean, I think doing something at this depth was important for this particular study, but it's not necessarily important going forward. But potentially a larger number of subjects, and maybe sampling lymphoid tissue as well as, and bone marrow as well as blood, and kind of looking at how things differ across those subsets, I think is really important. Yeah. And we're also going to try to check and see if what we're saying is actually true, right? So if you look at that model that we have at the end, if you look at kind of pre and pro B cells before they've kind of, as you step through the central tolerance selection processes, are you actually becoming more similar, like we think that is happening? So that's something we want to look at also.
Thank you very much. It was a fascinating talk. So I'm fascinated by this concept that once you sample those certain number of people at this depth, you stop seeing new things, and you get the leveling out of the number of new clonotypes you see. And so from a population genetics point of view, that makes a lot of sense to me, because it's sort of analogous to how you sample a large population, how the 1,000 Genomes Project was sampled. You sample large numbers of individuals at low depth rather than small number of individuals at very high depth. And so I wanted to sort of put that out as just a question that actually a lot of these studies are based on very small numbers of individuals for obvious reasons. Does that finding actually say you should be looking at much larger number of individuals at lower depth, which would probably help clarify some of the population genetics and allelic issues?
Yeah, so if you're not busy next month, I have a study section for you to be on.
Just to follow up on the same subject, the same question, just to say also about the ethnicity and, you know, those individuals, or like, you know, geographic location of those individuals as well, like, you know, if they're all Americans
again,
or...
So that gets back to the question, the comment that he had earlier, is that this is super limited in terms of number of subjects. So it was balanced. It was self-reported ethnicity. So half of them were Caucasian and half of them were self-reported African-American. Half were male, half were female. But they were all from like Southern California, right? So geographically, there's essentially no diversity. Ethnicity, there's some moderate diversity and matched kind of equal amounts of genders. But again, it's not great. So we need a much, like 1,000 subjects. Again, 1,000 subjects from Southern California might not be the best way to do it. 1,000 subjects that are spread, kind of a globally diverse population, maybe even spanning a range of ages. I don't know how many different variables you can put into one study like that, but yeah, absolutely.
To max out a bit later than in the 10, maybe 10, 11.
And especially, I believe it wasn't Corey because he wasn't here, but someone showed a plot of the haplotype diversity and how you have kind of outlier populations so that their IG locus is different. So we'd expect their repertoires to be, their expressed repertoires to be different as well. So yeah, absolutely. Good, thank you guys.
Thank you.